And we're
All
rolling.
right. Hello, everyone. Welcome to week 4 of PSPlatypus. Let's get right into it. Okay, so firstly, I'm covering epithelium today. So what we'll start with to contextualize epithelium. So we have 4 primary tissue types, 4 main types of tissue, and epithelium is one of them. And the other 3, connective tissue, muscle tissue, and nervous tissue, those will all be covered in upcoming weeks. All right. So what is epithelium? So epithelium is the tissue that covers or forms body surfaces. So this isn't only external surfaces like the skin, but also internal surfaces such as that lining the gastrointestinal tract, the cardiovascular system, so your veins and arteries and blood vessels, urinary tract, all of those and more. Its key features are that it's avascular, so it's not vascularized. It doesn't have blood vessels running through it. And that's because the underlying connective tissue underneath the epithelium is what supplies and deals with waste transport and things like that. There is very little space between the cells. They're strongly attached. They're bound together very strongly. They are oriented, so they have direction to them. And that's because they are related to covering a surface, right? So they need sort of a top that is facing the surface that they're covering, and a bottom that is lining the more interior aspects, internal aspects of the tissue. And they regenerate quickly because they're covering surfaces, and one of their main functions is protection. And so you can imagine through protection, they have to protect things that might be rubbed up against. So for instance, you graze your hand or something, right? You're going to need to regenerate your skin quickly to protect your more internal structures. Other functions of epithelium include absorption. So in the small intestines, epithelium will absorb food, but in the kidneys, epithelial cells will absorb blood. Epithelium also functions to secrete things in other parts of the body. So in the small intestines, it secretes enzymes. In the trachea, it secretes mucus. It also performs transport. So it lines vessels that can transport things. And specifically, the epithelium that lines blood vessels, that's called endothelium. It has a special name. And it also performs sensory functions. So the epidermis of the skin, for instance, that performs sensory perception. So that's how we can touch things and feel sensations such as vibration, heat, pain, and touch. All right. So epithelium, it is classified by the number of layers it has, the shape of the cells of the epithelial cells that make up those layers, and some additional factors that are slightly more specific. So to start off with, if you have a single layer of cells, such as over here, we've got squamous cells, but we've just got a single layer of these flat-shaped cells, then it's classified as simple. That's the same for cuboidal and the same for columnar. So you can see different shapes, but they're all a single layer. And so that's what we would call simple epithelium. Stratified epithelium is where we have whichever cell shape, but it is in multiple layers. So it's more than a single layer. So you can see here stratified squamous and stratified cuboidal cells. Then you have cell shape. So that's another key component. So if an epithelial cell is sort of flat or squat, then it is considered squamous. And that means, and that's just referring to its shape. And you'll see that in sort of like the skin, for example, is stratified squamous tissue. And cuboidal, well, it's like it sounds, it's more equal in width and height, or columnar, where it's taller than it is wide. Additionally, we have some more factors that contribute to the classification of epithelium. So we have keratinization, so that's keratin, which is a protein that you get in your skin, for instance, and it adds to the strength of the skin. So skin, for instance, the epidermis would be stratified squamous, and it would have keratin, a lot of keratin protein in these uppermost cells, which would also be actually dead skin cells. And they would be performing that protective function, especially. We also have transitional cells. So transitional epithelium is the type of, is where you have cell shape that can change depending on how much pressure is pushing on the cells and other factors. And that sort of makes sense in terms of, if you think of the bladder, as the bladder fills with urine, it needs to stretch. And so the cells that are lining the bladder would need to compress a bit to increase the volume of the bladder to store more urine. And then as the bladder is emptied, then the cells can widen out a bit more. And you also have pseudostratified. So you can see it's only one layer, but it appears like it's multiple layers. And you can see that here, this pseudostratified columnar epithelium. You can see there's actually only one layer, and that's usually the case with columnar cells. You don't really see pseudostratified with other types, in my experience. Okay, so as we were saying, epithelial cells are oriented, or they're polarized. They have top sides and bottom. And so this is here, you can see it's probably cuboidal, and it is simple. It's only one layer thick. You can see some microvilli up here and the basal lamina down here. So it's only a single layer of epithelium here, but it's just to illustrate the different sides, the different components of an epithelial cell. So you have the apical surface, and that's where you'll get things like microvilli and cilia. So that's to interact with the lumen, the outer space. You have lateral surface, which are the sides of the cells, and that's where you get a lot of lateral junctions that are these spooky diagrams here, but I'll go through them soon. And you have basal, which means like the basement membrane, also known as the basal lamina. And that's where you get some hemidesmosomes attaching to the basement membrane. So for the apical surface, that's facing the empty space. That's the top of the epithelial cell. And you can get different structures that are produced by the epithelial cells. So for instance, you can have microvilli. We have an electron micrograph here of some microvilli facing the lumen, the outer space. And they function to increase the surface area in intestine and kidneys, for instance, for absorption functions. And they use actin filaments that don't actually puncture through the cell. They're not sticking outside of the cell. They're inside of the cell, but they're poking in sort of the same way that you might imagine the poles in a tent would keep a tent quite pointed. And so they're stretching the plasma membrane of the cell out into that lumen through the use of actin filaments. Cilia, they are large, and generally they're motile, so they move stuff. So they, for instance, move mucus, they move sperm, and they move ova, so eggs. And that means that they require energy. They use ATP to do that. They have a 9+2 arrangement of, I think, microtubule doublets, and they are, they function. They are moving. That's how they do their jobs. And if you have cilia that are non-moving, well, there are some cilia that are non-moving, and they're called stereocilia, and those perform a sensory function rather than a function of moving things. Okay, we've moved from the top to the sides, the lateral surface, and we have four main lateral junctions to discuss here. So we have, I might just move this across. I'm not sure if that'll help, but hopefully. Okay, good. All right, so we have the zonular occludens. So zonular, take it meaning sort of a zone or a wider space, and occludens meaning to occlude, to stop, to block. So it's also known as a tight junction. You can see that depicted here in this picture. And that's designed to prevent transport of molecules between cells. Imagine it as a completely watertight fluid barrier. You can't transport things between plasma membranes. A zonular adherens, it's zonular, the same sort of thing. It's a wider space. And and adherens meaning to adhere, to stick, to bind. And it attaches cells to one another using actin filaments. You can see that depicted here. And it wraps around sides. So it's more of a zone than the next one that we're going to talk about, which is the macular adherens. Macular sort of meaning spot. And so that's a spot junction, also known as a desmosome. And that performs an attachment site, but in a more specific sort of spot rather than in these wider wrapping structures. And it uses intermediate filaments rather than actin filaments. And a gap junction, which is also known as a nexus, that actually helps transport. So it's sort of the opposite of a zonular occludens, if you think about it. I mean, that's depicted in 4 here on the diagram. It facilitates transport between cells. So it's sort of like a passageway, a tunnel between cells. And then we're onto the basal surface. So that's the bottom surface of an epithelial cell. And that's where you'll find at the very bottom layer of the epithelium. So if it's stratified, then you'll only get the basement membrane at the very bottom layer, separating the epithelium from more connective tissue. But what you'll get is the epithelium, and then you'll get the basement membrane. And that performs a function to adhere cells. So that basically performs a barrier between the connective tissue and the epithelium. And it adheres cells. And the way that it does that is through another sort of junction, similar to our desmosomes, but they're called hemidesmosomes. They bind epithelial cells to the basal lamina. And yeah, the basal lamina also helps maintain the orientation. So remembering that the epithelial cells are polarized, they're oriented, and the general shape of the epithelium, and it separates it. And an important fact is the fact that only cancer cells can cross the basal lamina. So you'll only see epithelial cells crossing the basal lamina if they've become cancerous. And this is just a comment about histology, because a lot of the way that you'll be interfacing with epithelium is through looking at microscopic images of epithelial tissue. And so I just wanted to give you a bit of advice on how to identify different types of epithelium. So a really important concept is the fact that the shape of an epithelial cell, as is the shape of any cell, it's related to its function. So we've evolved the shape of these certain cells in certain body tissues or certain spaces to perform specific functions. So for instance, squamous cells, they have a thin membrane, so they're good for absorption and diffusion. So that's why you'll find squamous blood cells in blood vessels and in the lungs. And they're also smaller volume and flatter, so they're less expensive to produce and they're more easily layered. And so that's for good areas where abrasion happens, where you've got a lot of friction and likely damage. So for instance, on the skin and in the mouth. And for instance, with cuboidal and columnar cells, they have a greater volume, and they have more capacity to produce mucus and hormones and things. So for that secretion function. And so that's why you'll find them in the gastrointestinal tract, for example. All right, thank you very much.
All right, thanks to Kyle. Do we have any questions for Kyle just straight off the bat? Remember, of course, you can chuck those in the chat at any point during the session. Oh, we got one.
I'll have a bit of a read up and answer in the chat. It's just been asked, how do transitional epithelial cells change shape? Where the signals to change shape come from? Also, why would you want to prevent transport of molecules via zonular occludens? I'll have a quick Google and give you an answer in the chat. Perhaps as we continue on with biotech.
No worries. Give me 1 second. Okie dokie. Can people say that? I'm going to assume so. Okay, so biotechnology. Now, this is basically just talking about recent technology. So like stuff that's come up over the past few years or relatively short period of time, which we use for various therapies. So this is all emerging technology or stuff which hasn't been used for too long, basically. So there are three types of technology that you actually need to know. And these are recombinant DNA technology and therapeutics. So in fact, I've broken my own rule immediately because this one is quite a bit older and we've been using it for a little while now to replace basically a variety of different technologies which we use to produce various proteins. We'll get into that a little bit more when we talk about it. Therapeutic cloning is still in its early stages, especially certain types of therapeutic cloning are still being developed, and gene therapy is, if anything, in its infancy. So we're very much on the cusp of developing that. You might have heard things about, say, CRISPR in the news, various points. That's really what we're developing now and trying to get to a state where we can actually use it regularly in therapy. Okie dokie. So for each of these, we need to understand the basic, like what are we actually trying to do? So what's the aim of this therapy? What are we trying to do by performing this therapy? And then we need to know an example of how this therapy might work. And we need to know what is the actual usefulness of this technology in a clinical setting. Okie dokie. So the first type that we're going to talk about is recombinant DNA. Recombinant DNA is basically the name for the technology that we use to make cells into protein factories. That's a very confusing explanation. It'll make a lot more sense as I go through it. So we're going to use a few tools to make these cells into our factories. The first one we're going to use is something called a restriction enzyme or an endonuclease. So these are basically bacterial enzymes which cut genes at a specific site. So they'll look for a certain sequence. You can see up there in the diagram to the top right. They're going to look for a certain sequence of nucleotides and then they cut at a certain place there. So if you think about it, they could either cut straight through. So in our example there, you can see it goes GGATCC. It might just cut through between the A and T in the middle, and that would leave a flat surface. Or, as you can see here, it's made sort of a crooked cut, and it leaves overhangs. So those are what we call blunt ends and sticky ends. Now, we don't care about the blunt ends because they're not actually very useful to us. But sticky ends, like the one you see here, are very useful because they make compatible ends out of different DNA molecules. So if you cut any piece of DNA with this restriction enzyme, it will have the same end because it's been cut at the same sequence and at the same position, which means that if you cut two completely random pieces of DNA with the enzyme, then you might be able to join them together. And that's exactly what we do. So we use DNA ligase. You might have heard of DNA ligase when we talked about DNA replication. So that's the one which puts our Okazaki fragments together. In this case, we're just going to use it to connect these two pieces of DNA after we bind them together using the sticky ends from the restriction enzyme. And a vector. So a vector is in disease, something which gets the disease into your body. In this, it's something which gets our genetic material into the cell that we're going to use. So plasmids are extremely common. If you did Bio 3-4, you probably heard about them. We can also use viruses, and we can also use just artificial genetic information. So the important things to remember is we need an origin of replication, which is somewhere where you can start transcribing genes from, basically. If you think about it, if we didn't have one, then of course, all the genes on there are just going to be junk DNA. They're never going to be actually used. We need a recognition site for the restriction enzyme. So if you look at that little sequence up there, top right, we need to have GGATCC somewhere. And otherwise we won't be able to cut the actual vector using the restriction enzyme. And we need to have a selectable marker. We'll talk a bit more about this in a sec, but basically it's something which is unique to this plasmid and will allow us to check if a cell has taken up the plasmid. So a very common variation of this is antibiotic resistance genes. Oh yeah, quick note about restriction enzymes. So in the natural world, they're found in bacteria, where they actually help break down viruses. So if a bacterial cell is infected with a certain virus, it might use a restriction enzyme to try and cut up the virus's genetic material. All right, so here's our actual sequence. So we need to first find our gene of interest. So let's say we're going to do a human growth hormone recombinant DNA test sequence. So we take the human growth hormone gene, we cut it using the restriction enzyme. We also cut the plasmid with the same restriction enzyme. So we need to pick a restriction enzyme which can cut both of them. And then we're going to be left with two pieces of DNA, the plasmid and the gene, which have compatible sticky ends. So we can put those together. They'll automatically bind together at those sticky ends because of the complementary shape. And then we use ligase to form covalent bonds between them to actually bind them together permanently. And then we need to transform the host cell. So the host cell can be a number of different cells. In this case, we're going to use an E. coli bacterium. And we can use heat, electric current, or another method. And basically what we're trying to do is we're trying to disrupt the cell membrane, because the cell membrane doesn't want to let the plasmid in. We need to force it to let the plasmid in. But once the plasmid is inside, then bacterial cells, mammalian cells, yeast cells, they will all start to transcribe that plasmid material. And then we're cooking. So we need to introduce the actual factor for that marker gene. So if we chose an antibiotic, we need to put that antibiotic into our little agar plate or whatever it is we're transforming things on. And so we kill everything that hasn't taken up the cell, hasn't taken up the plasmid, sorry. So that's what we did the selectable marker for, because we needed a way to test if it take it up, because none of this is foolproof. So we might not bind correctly at the plasmid stage. We might not take it up correctly at the transformation stage. So we need to have checks throughout the process to make sure that it actually happens as we intend. And then we culture the successful cells. We find all the successful cells, and then we grow them and we replicate them, because we want to make a lot of material, right? And we don't want to make just a little bit of whatever hormone we're trying to make. Okay, so there are a few different vectors, like I said. Sorry, this shouldn't say which vector to use. It should be which host cell to use. I'll fix that after this. But we can optimize for different things. So bacteria and yeast cells, they grow very quickly. They produce proteins very quickly. So if we want to make things fast, then we're going to use bacterial and yeast cells. However, if the protein that we're trying to make requires post-transcriptional modification, then we're going to need to use either a mammalian or a yeast cell, because the bacteria often just can't do that. So if you need to do something like you say add a sugar for glycosylation of a protein, then you need to use a mammalian or yeast cell. A bacterial cell just won't cut it. Mammalian cells are more expensive than the other two, but they can handle large proteins better. So if you're making a big protein, that might be the way to go. Like I said, lots of options. It's a case of what you want to optimize for. All right, there's a little aside. We can do something called farming. So instead of culturing cells in vitro, so this is when we do it in a lab, in a test tube or something, this time we put the vector into an embryo, and then we implant the embryo into an animal. It's then born as its own animal, and that offspring basically serves as our transformed cell, except it's got millions of cells, and many of them are going to be expressing the protein that we want. So in this case, we're using sheep, and we're going to take their milk basically, and it's going to contain a protein that we wanted to be expressed, and so we artificially inserted it while the sheep was still an embryo. And so it's not quite as controlled as a lab environment. So in a lab environment, we can pretty much control everything that's happening. In this one, it's a living animal. There's a lot more things that are going on. And so we need to purify the protein. Okie dokie. So there's another process that we need to talk about, which is basically, okay, we have a gene of interest that we need to get in. How do we actually get that gene of interest in significant quantities? Because we don't want just one gene of interest. We're going to want thousands of these, because we want to have thousands of cells. And so we use a process called the polymerase chain reaction. And this is basically, we double a DNA sample over and over again until we have a huge amount of it. So it's basically a three-step process, which we repeat again and again and again. So in the first step, we heat the double-stranded DNA. And once it reaches about 94 degrees, that's going to separate the strands. So you remember when we were doing DNA replication in the first place, when we heat it up, the hydrogen bonds between those two strands are going to break and they're going to drift apart. Then if we cool it down not quite enough to make them rebind, but we cool it enough to put these little primer strands, the little sequences of single-stranded DNA, which will attach to the 3' ends. So we make sure with the primer strands that we make them very specific. They'll only attach to the 3' ends of the sample. And then we heat it up again, and we add not a human DNA polymerase, but we add a heat-resistant Taq polymerase, which comes from a bacteria, to make a new strand. So we've started with a double strand, we've then separated into single strands, and then we're going to duplicate the single strands so that we end up with 2 double-stranded samples. So we've effectively doubled the DNA. And yeah, so we use Taq polymerase, which comes from like the bacteria which live around hot springs. We use it because they're used to higher temperatures, and higher temperatures give us a faster speed of replication. We don't want to be waiting too long. Again, not perfect, but we can make it pretty reliable. So in practice, this will be, you chuck it in a machine, you chuck in your sample, your Taq polymerase, free nucleotides to be used, and your little primers. You put them all in a mixture, and you just cycle the machine again and again. And basically all it does is it changes the heat, and that drives the whole process. Okay, so this is a diagram, basically. You can see we start with our double-stranded DNA. We then pop out our primers. The primers help build up a new double strand, and then we do it again. And so although we only have 2 original strands here, we're going to have pretty good copies for all the other ones. Now, one thing you might notice is that it's not quite the same as the beginning. You'll notice all of the new strands are slightly truncated because the primers don't always bind right at the very end. And so we might lose a little bit of genetic material in the end. So generally, you're not going to only have your gene of interest in the sample. You'll have your gene of interest plus a little bit of junk DNA to either side to give yourself a bit of a buffer. Okie dokie. So here are some of the therapeutics that we can make. You don't need to memorize this. A couple good ones to know would be things like growth hormone and insulin. And one that has come up sometimes is tissue plasminogen activator. So you guys should already know what growth hormone and insulin do. TPA is basically used to prevent blood clotting. So yeah, again, important thing to understand here, there are proteins because we're using genes. Of course, genes translate to proteins. And we're not creating lipids, we're not creating steroid hormones. We can only create things which fall within protein and peptide bounds. Okie dokie. So the second technology you need to know about is called therapeutic cloning. So we're not trying to make an organism, that would be reproductive cloning. In therapeutic cloning, we're trying to make basically a whole bunch of cells, which are the patient cells, but we're trying to make patient-specific stem cells, okay? So we have 3 classifications here for the ability of cells to differentiate, basically. So right when you're born, very soon after conception, you have totipotent cells. So they can make literally any cell in the human bodies and stuff which isn't in the human body, so like the placenta. So this can give rise to literally anything. Then a little while later, you're going to specialize a tiny little bit, and lots of your embryonic cells, when you're little growing in your mother's womb, are going to be pluripotent cells. So these can make anything in your body, not the extra stuff. So we don't usually use totipotent cells because we don't need those extra embryonic tissues. We're not trying to make a baby, we're trying to make usually organs or specialized tissues for the patient. So pluripotent does pretty fine for us. We also have multipotent cells. So these can be usable if we convert them later, but we can't use them straight because they can only give rise to cells in their own little category. We want to have cells which are able to make a wide variety of things because we need to make tissue for the patient. And so the other question is, why do we care that the cell line is specific to the patient? Why not just give them tissue from anywhere? And the answer, of course, is that we need to make sure that the patient's body doesn't reject this new tissue. And the easiest way to do that is to make sure it is self-tissue. All right, so the first method of therapeutic cloning we have is called somatic cell nucleotransfer, usually SCNT abbreviated. And so we take an egg, we take the nucleus out of the egg. So you remember the nucleus is a haploid, only one N. Then we insert a nucleus from a mature cell, which is going to be diploid. So if you remember, during the process of fertilization, we have an egg, haploid, sperm haploid. They bind together and form a diploid embryo. So in this process, we instead achieve diploid straight by getting a nucleus from a mature cell. So we artificially stimulate that to make it into an embryo, and that will basically allow us to create stem cells. So these stem cells are they're proper embryonic stem cells, so they are literally from an embryo, and we can make them into many different cell types. These are pluripotent cells. Okie dokie. Now, we don't only have to use natural pluripotent stem cells, we can also use unnatural pluripotent cells or induced pluripotent cells. Induced pluripotent stem cells, can't speak today. All right, so this is where we, instead of taking the adult cell and just cribbing its nucleus, we take the whole adult. And instead of putting it into embryo to make it into stem cells, we target a set of 4 genes which in normal adult cells turn off the ability to be pluripotent, basically. And instead we flip those genetic switches, and now they're pluripotent stem cells. So we've sort of turned back time a little bit in these cells and made them think they're more embryonic than they actually are. So it's pretty good, but it's not perfect. And so there are a few little discrepancies, basically, inconsistencies between an actual embryonic stem cell and an induced pluripotent stem cell. You can see them there. So they're not quite perfect, but they're pretty good. Now, basically, the reason we would bother using a less effective method, which is, as we mentioned, iPSC has a lot of practical problems. SCNT is a lot more reliable, but it has ethical issues. So you might have heard about like the ethics of, should we be making embryos and then destroying them immediately to harvest the stem cells? Is that the right thing to do? And so that's an ongoing question, and iPSC sort of dodges those ethical questions. But in the end, we're trying to do the same thing. So in both cases, we're trying to get pluripotent stem cells, which we can use to make cells for the patient, which won't be rejected by them. Okie dokie, our third and final therapy. This is by far the newest one and the least well understood one. But this is basically where in the previous therapies we modified cells outside the patient. In this one, we're going to change the patient's cells while they're in the patient. So obviously it's a lot more involved than the other types we have. So right now, the only thing we can do is we can add a functional allele into a patient with 2 defective alleles. So what that means really is you might have, say, if you guys have learned about cystic fibrosis, I don't know if you have, then you might have 2 defective cystic fibrosis genes, all right? So you're not able to make the protein that healthy people can make. So we can help you by putting in a working allele. So if you have the allele somewhere for a cystic fibrosis gene, then that'll help you make the correct protein. And so that is effective gene therapy in its own right, but it's not on the level of actually fixing the gene in the patient. And there are more errors there. So like if we use a viral vector, which is one of the ways that we can currently do it, we might just have an immune response against it. We can have cancer, and we can have degradation over time because this is still foreign DNA that we're introducing. Right, so CRISPR-Cas9, this is the interesting one. This is the new one, which you've probably heard of. And so this is basically where we use another bacterial enzyme. We use a lot of bacterial enzymes, they're quite useful to us. But this is where we use one to correct genes. So this is the one where we're trying to actually edit genes properly while they're in a person. So a little background for CRISPR-Cas9. So this is another case of a bacterial enzyme which was initially meant to defend against viruses. So in this case, it's you give it a little bit of RNA. In a bacteria, that bit of guide RNA that we have is going to be complementary to a viral DNA sequence. So CRISPR-Cas9 is meant to find a virus using that guide RNA and then basically cut the virus's DNA, prevent it from actually functioning properly. So in this case, we actually make our own guide RNA. So we make our own guide RNA. We make it complementary not to a viral DNA or RNA sequence, but we make it complementary to part of the human chromosome, the specific gene that we want to actually edit. So we give it that guide RNA, and Cas9 already knows to follow the guide RNA somewhere and then cut. That's what it knows how to do. It needs to follow and cut. So if we give it a guide which will take it to a certain place in the human chromosome that we want to cut, it's just going to do that. automatically. So we've successfully made a cut there. Now current the current more reliable thing we can do is basically knock out a gene. So we can prevent a gene from functioning. And the easiest way to do that is basically just to cut. And although the body does an okay job of repairing things, it also sometimes screws up. So you probably end up with, say, 1 or 2 nucleotides at least, which are different, and that it could very well be enough to inactivate that gene. And so basically our strategy here is based around screw things up a little bit, hope that the body can't fix it properly, and that'll knock out a gene for us. Now, the more ambitious strategy is to try and knock in a gene. So we give it a piece of, we add a piece of correct DNA, and we try to cut it and then put that in. Excuse me. So that is the general idea. That is what we're trying to build towards, because that will be, if we are able to do that, then that has really, really promising implications for gene therapy, because then we can actually start to think about, okay, well, this patient has 2 defective alleles. We can actually just straight up fix in there. Of course, this is, again, it is a very early stage, and we have a lot more things to think about, like, for example, scalability. We need to be able to actually use this on a wide scale, because your person, like you, has millions of cells in your body. We need to edit a lot of them, if not all of them. Of course, it also has its own dangers. None of this is foolproof. Okie dokie, do we have any immediate questions based on that? I'm just gonna have a look through the chat. Okay, so somebody asked, multipotent only give rise to their own cells. Right, okay. So that's usually a little bit more specific than ectoderm, endoderm, and mesoderm. So you'll have in your adult body, you will have stem cells. You still have cells that are capable of differentiating into a wide variety of cells. It's just a case of that variety is less than in an embryo. Okay. So you'll have cells in your nervous system, you'll have cells in your muscle, in various other places in your body, which are capable of producing cells as needed. And of course, especially in your bone marrow and whatnot, you have the ability to produce heaps and heaps of red blood cells and that sort of thing. Yeah, so note that red blood cells don't have a nucleus. They're not going to be able to replicate themselves. They need to come from other cells. So we have mechanisms in the adult body to create other cells, but we don't want that. We want to create cells that we can make any type of tissue from. And to do that, we need stem cells. Okie dokie. Feel free to chuck anything else you guys have to ask in the chat. I'm going to move on to liver metabolism right now. So the liver, basically, it's able to carry out a bunch of specialized functions. This is like, not only does it do a lot of the stuff that is done all over the body, it's just very good at it. It also does certain things which only the liver can do. So when we have more energy than we need, we have an excess of energy, we can store it through the liver, in the liver, or we can have conversions happen at the liver. So when we want to store it as carbohydrate, we store it as glycogen. So we're going to have a whole bunch of different terms here which sound very simple, so pay attention to these ones. This is glycogenesis. That's when we're making glycogen. So liver and muscle cells store a lot of this. So muscle cells because they need energy reserves to function, and liver cells because, as we said, the liver is sort of one of the biggest components of metabolism. And when we want to store things as lipids, we turn them into TAGs. That's our preferred method of storing things. And we can again, remember we've talked about acetyl-CoA as sort of a centerpiece of metabolism. So it is sort of the universal step that we can use to convert things from one type to another. So we can convert fatty acids into carbohydrates, and we can convert carbohydrates into fatty acids using the acetyl-CoA sort of intermediary there. So in this case, we turn carbohydrates into fatty acids, and then we combine them with glycerol to make our little TAGs. So the benefit of this is that triglycerides have a much higher energy density, basically. So they're harder to get energy out of when we actually need to mobilize it, but we're able to store a lot more efficiently. So that's why your body builds up fat, basically. And also, sorry, I don't know if you know, but certain cells in your body will also require either type. So certain cells in your body need carbohydrates, certain cells in your body need lipids. Okay. So so this is quite important actually. So when we store energy as the glycogen, it behaves differently upon breaking it down again, depending on which cell in the body it is. So the immediate comparison that we make in medicine year one is between the liver cells and skeletal muscle cells. So if you look at this diagram, ignore the little blue lines. I couldn't find a way to get rid of them. Excuse me. When we break it down, it actually comes out as glucose 6-phosphate, which is a little bit different to glucose, enough to be distinct. And the main like functional difference between glucose 6-phosphate and glucose is that G6P can't go in the bloodstream. It can only be within the cells, and glucose can go in the bloodstream. So our liver cells are able to take glycogen, break it down into G6P, and then break it down further into glucose, and then put that into the bloodstream. So when we want to mobilize sugar and actually increase our blood sugar, then we need to call the liver, because these are the ones that are specialized to be able to actually send our glucose all throughout the body. Muscle cells, on the other hand, when they break down glycogen, they can only use glucose on site. So they have to be the ones using the glucose. And before you get any ideas, when we have glucose in the bloodstream, whenever it enters the cell, it gets converted into G6P. So it's a one-way process, except in the liver. Once the glucose enters the cell, the only way it usually leaves the cell is by metabolizing. Okie dokie. So we also have a process called gluconeogenesis. So this is basically where we create glucose out of other things. So I mentioned acetyl-CoA there. We can convert acetyl-CoA into pyruvate and pyruvate into acetyl-CoA. And so gluconeogenesis starts at pyruvate and ends at glucose. But we can also use it as more of an umbrella term to say, okay, when we want to take amino acids or glycerol into glucose, we say that's gluconeogenesis. We are making glucose out of other things. That's why we have the neogenesis part. So yeah, so this is sort of the opposite of glycolysis, but it's not the same steps. So like avoid saying it's just glycolysis in reverse, because it's not. There are little differences between what is actually happening. But the main point is we're in one process, we are breaking down glucose in order to release energy. In the other one, we are making glucose to then be broken down later in certain cells. So like I said before, certain cells in your body need certain types of fuel, and brain cells are one of them. So this is really important because obviously you need to maintain energy flow to your brain. And the only way to do that is to keep your glucose up in your bloodstream, because lipids can't cross what we call the blood-brain barrier. And so we need glucose to act if we actually want to keep the brain fueled up. Now, we also use this process to recycle lactate. So when we ferment, when we ferment pyruvate instead of actually going through aerobic respiration, then we can end up with lactic acid buildup in your muscles. And so your body doesn't like that being around, so it needs to get rid of it. You probably would have heard this in Bio 3-4. What you didn't get taught was how it gets rid of it. So we use something called the Core cycle. I'll roll it back just a sec. So you can see here, when we ferment pyruvate, look at the skeletal muscle cell on the right here. When we ferment pyruvate, it turns to lactate, and that lactate goes to the liver and is then turned back into pyruvate, and we can turn it back into glucose. So of course, this actually requires energy. So we need energy to turn it back into pyruvate and glucose. And so it's not necessarily we do this to get more energy. We do this because we don't want lactic acid hanging around, basically. Okie dokie. Any questions of that? See something in the chat. Do we need to know every step of certain process as glycolysis? Please, God, do not memorize every step of glycolysis. That is a bad idea. You definitely don't need to know every step of glycolysis. You don't need to know every step of the Krebs cycle. For Krebs, it can be useful to know certain intermediates. So things like oxaloacetate can be useful just knowing like certain steps around there, because they're useful when we're converting substances from one type of macromolecule to another. So, when we're converting between amino acids, lipids, proteins, all of that, it's good to know what the intermediate steps are. But you don't need to know the step in the Krebs cycle. You definitely don't need to know the step in glycolysis. They're like 12 steps or something, and they're not really useful at all to you. Like literally our lecturers will say to us, what is the most useless thing I learned in med school? The Krebs cycle. So you need to know how it works. You need to know how we can get things in the Krebs cycle, but you don't need to know the steps in the Krebs cycle super well. All right, over to Raymond.
Too easy. Wow. Now that you've learned a bunch about the processes, we're going to have a look into at what points these processes are used to create energy and why it's preferenced in such a way. So in terms of adipose tissue, it's composed of adipocytes, and they are primarily responsible for storing triglycerides. This process is metabolically activated. So after the meal, fatty acids are delivered via chylomicrons stored as triglycerols. And in between meals, hormones signal the need for energy, thus mobilizing these triglycerides. The glycerol that is left behind during the mobilization goes to the liver to undergo gluconeogenesis to create more energy. And as such, 95% of the energy used by the body comes from beta oxidation, and only 5% of glucose. This is because beta oxidation is just a much more efficient method of producing energy, as one palmitate acid can produce up to 126 ATP. In terms of skeletal muscle use, at rest, muscles only consume 20% of total oxygen, and during activity, it can account for up to 90%. And this is, of course, understandable, as the more you exercise and move, the more energy expenditure you use, thus you need more oxygen to create the energy required. Muscle cells are specialized to produce ATP and can utilize any fuel source, and has stores of both glycogen and fatty acids. At rest, 80% or more of energy comes from fatty acids through beta oxidation, as this is, although a slower process, it is the most effective process to create energy. During moderately active stages, glucose is used primarily from glycogen stores, which stays as glucose 6-phosphate. All the liver and fatty acids and ketones can also be used, but the primary source of glucose. During short bursts, ATP reserves, which are just ATP that's already in the muscle cells, are used as it's a direct energy source, or creatine phosphate, which is just a molecule that is made from phosphates and creatine, and it's cleaved to create just creatine, and the energy that's produced via breaking that bond is used to cause muscle contraction, and this is an extremely fast process, so allowing for rapid energy use, so only used in short bursts. During moderately active stages, oxidation of glucose from the liver via aerobic respiration, as well as ketone and glycogen stores, as well as fatty acids, are used in the background, but the major one is just glucose. During extremely active stages, stored glycogen is the main fuel source, and it undergoes glycolysis, and then becomes lactic acid, as we're going via the anaerobic pathway, as it can create energy quickly. And this is because the body, the muscle cells are lacking oxygen, as the heart rate and the respiratory rate need to first start up and catch up to give enough oxygen to the muscles to actually begin aerobic respiration. During a state of starvation, your body has run out of your fats as well as your glucose glycogen stores, so it's left with only protein, which is broken down into amino acids and can be converted into glucose through gluconeogenesis in the liver. And this is because this is extremely important, as some parts of your body can only use glucose, such as the brain. This is a nice schematic here that shows you what is being used as exercise begins. So in the first few seconds of exercise, your ATP and creatine phosphate is used. As we progress, we start burning anaerobically, so you're through the fermentation pathway to create energy. And then at this, after a few more minutes, your respiratory rate and cardiac rate increase to a point that aerobic respiration can occur, therefore resulting in aerobic metabolism. And then the diagram on the right showcases how fats are the primary source at the beginning, they die down, and carbohydrates ramp up, and then it switches over at about 30 minutes. The heart muscles are continuously active and cannot undergo anaerobic respiration, and also the heart has no energy storage. The heart's primary use is fatty acids, especially after eating. After a long time, the heart can also use glucose from the liver or ketones. As the brain has a highly selective barrier known as the blood-brain barrier, only glucose and ketones are able to cross it to provide energy to the brain. And glucose is the primary source and ketones are the secondary source used during starvation. And glucose usually undergoes the aerobic pathway, and during starvation, the brain will use ketones under the condition. This allows for the brain to maintain its energy sources even in extreme conditions. Red blood cells have no organelles, as their just primary function is to carry oxygen and no mitochondria. So the only way that it can produce energy is through the anaerobic pathway. And as such, red blood cells purely rely on glucose, and they can only undergo anaerobic respiration to get energy. And here's a quick summary that outlines the specific details of what muscles and specific cell types in the body use as their main energy source, as well as in the states, different states. Awesome.
I just have a quick question, sorry.
Shoot.
Is ketone bodies the primary source for energy during starvation?
Or
Or is it like storage of glycogen and it's first converting it to glucose, and that it relies on glucose first?
it always relies on glucose first. Ketone is like at the very extreme stages when we have burnt out of all the other carbohydrates as well as fatty acids present.
Sure. Yeah. And another question with the Great Metabolic Race.
Yep.
We did that last week. Between 0 and 5 minutes, is lactate ever used?
Ooh, I need to double check that.
Sure.
I'll have a check.
Could you restate the question?
Thank you. The question was that because what we learned is like between 0 and 10 seconds, you use creatine, creatine phosphate.
Mm-hmm.
And then they went straight after 5 minutes. But between 10 seconds and 5 minutes, is lactate used for an energy source?
What do you mean when you say lactate use? So lactate,
like, because you know how they have initially when you start racing, there's a lack of oxygen in the body.
Yep.
So does that mean that it starts fermenting?
Yeah. Okay. So can you see that green line, anaerobic metabolism? Yep.
Yep.
Yep. So that is anaerobic. Well, I mean, it's anaerobic metabolism. It's fermentation. So that produces lactate. So lactate is a product of anaerobic metabolism. We don't use it to create energy. So that whole Cori cycle process I told you, when we want to change lactic acid into something else, typically pyruvate and then to glucose, it actually takes energy. So if we were to change lactose to something, we'd actually be losing energy. So we'll be creating a whole bunch of lactate during this, but we can't use it as an energy source because it's not an energy source.
Right. And when we have that production of lactate, that's when our muscles start hurting.
Yeah.
Okay, cool. Thank you.
Too easy.
Hello guys. Now we're gonna talk about health enhancement program, which is a program for medical students to increase the awareness of their mental health, their well-being, and mindfulness. And it actually have shown that this program uh, can have many benefits, including improved immune function, reduce stress, anxiety, and depression. Also, increase the sensitivity for self pace and patience. And it also can improve the academic performance because always mental state is connected to your um, performance. So we have a little bit of terminology here. Um, Integrative medicine, it's a holistic approach to medicine that uses conventional medicine, evidence-based and safe therapies. So it doesn't just focus on conventional medicine, it also involves some alternative therapies, evidence-based therapies. Then we have the orthodox medicine, which is the conventional medicine, and it's generally what you learn during your medical school, and it's widely accepted in clinical practice. Some people call it scientific medicine because it's based on science, and everyone should use it. But this is not always the case because there are some not evidence-based therapies in orthodox medicine. Then we have unorthodox medicine or traditional medicine, and this is generally not what you will learn during your medical school, and it's not widely accepted in clinical practice. Then we have the complementary medicine, and as its name suggests, it's can be used as a complement for the conventional medicine to to minimize the side effects, the symptoms, reduce the cost, and enhance the outcomes. Lastly, we have alternative medicine. Uh, so this can be used as an alternative to the conventional medicine. So it replaces the conventional medicine. And as a result, we have complementary and alternative medicine, CAM, and this includes a wide variety of therapies. Some of them are good ones, some of them should not be used, but always the best ones are the evidence-based, safe, effective, and reasonable cost therapies. And nowadays, it's very important to know about it, to have a good idea about CAM, because more and more people are using it. You as a doctor, you need to know about what alternative medicine or therapies they might be used, because it might have some interactions with the conventional treatment. Also because more and more people using it, and you have a better knowledge about it, so there will be a better communication between you and your patient. Also, it helps patients to make informed and safe decision, because if there's alternative medicine other than conventional medicine that the patient can use, and it offers it's evidence-based and it's safe and reduce the cost, so the patient might need to know about it, or you need to suggest it to your patient. And finally, it's increasingly expected by the AMA, the Australian Medical Association. It has some advantages. It's more traditional, natural, holistic, and might have less side effects. However, it can cause some danger by having some side effects, especially if it wasn't evidence-based therapies. It can provide false hope for patient because it might just doesn't work. And finally, the poor regulation of its practitioners and some ethical problems regarding the informed constant. Then we have the modalities of CAM. So we have 4 of them: mind-body medicine, biologically based medicine, manipulative and body-based medicine, and finally, energy medicine. So mind-body medicine, it's based on the idea that anything can improve or change your mental state, will change your behavior and your physiology. Some examples of mind-body medicine: relaxation, meditation, and biofeedback. Then biologically based medicine, which is the usage of biological compounds that can be taken or applied. Some essential oils, aromatherapy, some supplements, and vitamins. Then we have manipulative and body-based medicine. So it's involves physical manipulation. It's like massage, osteopathy, and reflexology. Finally, you have energy medicine. So it's involved energy manipulation, and it has 2 main types: biofield therapies and bioenergetic therapies. We have this essence model, which is the basis of health enhancement program. So the program focuses on these 7 fields. It's an acronym, stands for E for education, to provide a better understanding of ourselves and our minds. Then S stands for stress. It's about mindfulness. S also for spirituality, which is the meaning and purpose of life. A for exercise and for nutrition. C for connectedness and our social relationships. And finally, F for the environment. So we have a preventive medicine, which is medicine focuses on preventing the disease from occur from the first place rather than treating the diseases. And we have 3 levels of prevention. We have the primary prevention, where we have a healthy patient with no disease, and this primary prevention is about to stop the disease progress before it's established in the healthy person. This can be achieved by lifestyle modifications, changing their lifestyles to be a healthy lifestyle, also managing risk factors. It's the most effective, it's the cheapest type of prevention. It's the minimize suffering, and but also it's the most difficult one, because it's all about changing your lifestyle into a healthy lifestyle and managing risk factors. Then we have the secondary prevention. So now the disease has occurred, but it's asymptomatic, but disease process will underway. This can be achieved by an early intervention and slowing the progression of the disease. This can be achieved by a lifestyle modifications and treating early stages of this disease. Finally, we have the tertiary prevention. Now we already have complications. Chronic disease has been established, and this can be achieved by providing rehabilitation to the patient and trying to prevent further complications of this disease. This one is the most expensive and the least benefits. now we have these acronyms that we can use to support us in the process of behavior change. We have firstly SMART acronym for goal setting. So S for specific. For example, if we want to do more exercises, so we need to have a specific goal. What's this goal? Why are we doing it? For the exercising example, you can't just say, I need to do more exercises. Like, you have to be more specific so that you can achieve your goal. You might say, I want to exercise, I want to run for 5 kilometers a day. Then your goal must be measurable. So in this example, like 5 kilometers a day is our goal. Through this measurement, we can see how far did we go in achieving our goal. It's also need to be attractive to encourage us to continue. For example, we can take some of our friends or our families during our exercise, so it won't be boring. Also, it needs to be realistic. So you can't go from not exercising at all to running 5 kilometers every day, every week. It's just very hard to achieve. It also needs to be timely. Like I need to achieve this goal in 5 months. Like if you put it in frame time, it would be better. Then we have the basic behavior change. B for behavior, what we do. A for the attitude or the effect. It's about your beliefs, your emotions. Then S for skills or sensation. It's about what can we do. And finally, K for knowledge, what we know about this behavior. Finally, we have the ADAPT acronym. This one for changing habits. So A stands for awareness. You need to be aware of your habit, like this habit is not healthy and I need to change it. Then D, decision. You will make a decision to change this habit. E for effort. Changing habits is not easy. It's like going uphill. So you need to put some effort in order to change this habit. P for perseverance. Just to have commitment, a commitment to changing this habit. And finally, T for tolerance of discomfort, like changing habits will not be comfortable for you, so you need to have tolerance to change that. Finally, we have Prohaska-Di Clemente cycle of behavior change. So these are the stages if you want to change your behavior. And you as a doctor might want to use it with a patient that, for example, needs to change a habit or a behavior. So we start with the pre-contemplation stage, where the patient did not think about changing their behavior yet. If we have smoking habits, we have smoker, he or she think that smoking is a good habit and it's not, it doesn't need to be changed. Then the contemplation stage where they start to think about changing this behavior. For example, if their doctor told them that smoking is harmful for you, it can cause lung cancer, for example, then they will think about changing this behavior. Then we have the preparation stage, which is a very, very important stage, and lots of people forget about it. This stage where you make a concrete plan to change your behavior, for example, searching about this habit, what are the disadvantages of this habit, and what benefits you will get from changing this habit. Also, you might want to inform your family or your close friends that you want to cut this habit or be away from people who, like smokers, for example, so that you won't be triggered by them when they smoke. Then we have the action stage, and this is where change in behavior occurs. This is when you or the patient cut the smoking. Then we have two pathways. Patient might relapse and smoke again. We need to think that relapse is not a failure, but it might be something that the patient can learn from what was wrong in this, what caused the relapse, so that they can avoid it next time. And when patient relapse, they must start again from the pre-contemplation stage, or the behavior will be successfully changed, and this is the maintenance stage. And that's it. Thank you so much.
All right, big thanks to Marlon. Do you have any questions about that or anything we've mentioned today? All good. Looks like we have nothing immediate. So with that, have a good day. See you all. Thanks for coming. We'll be here next week. Revision quiz will be out sometime later this week. Stick around and ask us if you have any questions. Have a good one.